Welcome back to Med Studios. Today, we are opening our histological field guide to explore the fundamental building blocks of the human mind, neurons. At a magnification of 400x stained with fluorescent markers, the microscopic architecture of these cells is nothing short of breathtaking. A neuron is not just a simple cell. It is a highly specialized electrochemical processing unit designed to transmit data across vast biological distances in fractions of a second. In this comprehensive guide, we will map out the complete anatomical overview of the neuron, from the central soma out to the farthest dendritic spines and axonal terminals. We will classify them by their functional configurations, how they act as sensory receivers, motor effectors, or critical interneurons. And most importantly, we won't just look at healthy tissue. We will apply this knowledge to clinical pathology. By the end of this video, you will understand exactly how microscopic neurodegeneration, demyelination, and synaptic dysfunction lead to devastating systemic diseases. Let's begin our journey into the nervous system.
Before we look at the physical structure of a neuron, we must understand its functional configuration. The nervous system operates as a massive, high-speed biological highway, with traffic flowing in two primary directions. First, we have the afferent, or sensory, pathway. These neurons act as the body's alarm system, carrying vital sensory information from the peripheral tissues directly back to the brain and spinal cord. For example, if you touch a hot stove, an afferent impulse instantly transmits that pain signal centrally. Second, we have the efferent, or motor, pathway. This is the command sequence. Efferent neurons carry electrochemical instructions from the central nervous system outwards to the peripheral tissues, such as stimulating a skeletal muscle to contract so you can pull your hand away from that hot stove. Bridging the gap between these two are association neurons, or interneurons, which act as the central processing hubs, entirely confined within the central nervous system, making the complex decisions between sensation and action.
When we examine the physical structure of a typical neuron, we divide it into three highly specialized functional zones. Section 1 is the receivers, consisting of the dendrites. These are highly branched tree-like structures that project outward to capture incoming electrochemical impulses and conduct them centrally. They are covered in microscopic bumps called dendritic spines, which form synaptic connections with other cells. Section 2 is the processor, also known as the cell body, soma, or perikaryon. This is the metabolic heart of the cell, housing the nucleus and all the vital organelles within its cytoplasmic membrane. It processes the incoming signals from the dendrites and determines if a new signal should be fired. Finally, Section 3 is the transmitter, the axon. This is a singular, elongated projection that transfers the newly processed electrochemical information away from the soma, delivering the final message to other neurons, glands, or muscle fibers across the body.
Let's zoom in on the processor, the soma. Right in the center, we find the blueprint, an unusually large, pale-staining nucleus containing a dark, prominent nucleolus. This massive nucleus houses the DNA and signifies that the neuron is a remarkably active cell requiring constant transcription. Surrounding the nucleus in the cytoplasm, we find the assembly lines, known histologically as Nissl bodies. If you look at a slide stained with a basic dye, Nissl bodies appear as dense, granular, dark blue clumps scattered throughout the soma. But what are they actually? They are massive collections of rough endoplasmic reticulum and free ribosomes. This heavy presence of Nissl bodies is a definitive histological hallmark. It proves that the neuron operates at an exceptionally high state of continuous synthetic activity, constantly manufacturing the massive volume of structural proteins and neurotransmitters required to maintain the long axon and facilitate continuous synaptic communication.
As we move from the busy soma toward the axon, we hit a critical transition zone, the axon hillock. Visually, the axon hillock is a highly distinct,
pyramid-shaped or cone-like funnel where the single axon originates from the dilated cell body. It is histologically notable because it is entirely devoid of those dark-staining Nissl bodies we just discussed, giving it a pale, clear appearance under the microscope. But its true importance is functional. The axon hillock serves as the neuron's primary signal integration zone. It is the ultimate biological calculator. It collects all the excitatory and inhibitory electrical signals funneling in from the vast dendritic tree and the soma. If the combined electrical charge reaches a specific critical threshold at the axon hillock, it triggers the firing of a brand new action potential. Therefore, the axon hillock is the exact initiation point where the electrical signal commits to traveling down the axon highway.
For medical and biology students, distinguishing an axon from a dendrite on a microscopic slide can be challenging. Let's look at the diagnostic table. First, consider length. Dendrites are relatively short and remain near the soma, while an axon can be incredibly long, sometimes exceeding 1 meter, like the sciatic nerve running down your leg. Second, examine the course. Dendrites bend, twist, and taper as they branch out to meet incoming signals. Axons, however, run a remarkably straight, uniform course with a consistent diameter. Third, look at branching. Dendrites branch early and often, creating a massive, complex dendritic tree to maximize their receptive surface area. Axons typically do not branch until the very end, restricting their branching strictly to the distal terminal arbors. Finally, check the origin. A single neuron has multiple dendrites originating from various points on the soma surface, but it only ever has one distinct single axon originating specifically from the clear, pyramid-shaped axon hillock.
Now, let's examine how these axons travel through the body by looking at a cross-section of a peripheral nerve. When you look at a standard H&E stained slide, you will notice a fascinating histological phenomenon we call the myelin illusion. Surrounding the dark central dot of the axon, you will see a large, empty white ring bounded by the thin endoneurium connective tissue. This empty space is actually an artifact of the slide preparation process. In biological reality, this space is densely packed with myelin, a thick, lipid-rich insulating sheath wrapped tightly around the axon. However, because myelin is primarily composed of fats, the harsh alcohol washes used during standard histological slide preparation completely dissolve the lipid layers. Therefore, the fat is washed away, leaving behind only the deceptive empty white space. Recognizing this alcohol wash artifact is crucial for correctly interpreting nerve tissue under a light microscope.
Why is this myelin sheath so important? It all comes down to speed and efficiency. Let's compare a myelinated axon to an unmyelinated one. A myelinated axon is wrapped in a thick, segmented insulating layer provided by Schwann cells in the peripheral nervous system or oligodendrocytes in the CNS. This insulation prevents the electrical charge from leaking out, allowing the action potential to rapidly jump from node to node in a process called saltatory conduction. This drastically increases transmission velocity. Conversely, unmyelinated axons lack this thick, segmented wrap. While they are still supported by a thin cluster boundary of Schwann cell cytoplasm, the lack of true lipid insulation means the electrical impulse must travel continuously down the entire length of the axon membrane. Consequently, the transmission speed in unmyelinated fibers is significantly lower. Therefore, myelination dictates both the structural thickness of the nerve fiber and the ultimate velocity of its electrochemical impulse.
Neurons are not all built the same. Their structural configuration is perfectly adapted to their specific role. Let's compare the multipolar and bipolar configurations. The multipolar neuron is the standard. It features a massive, complex dendritic tree extending from the soma and a single axon. This is the most common configuration in the human body, overwhelmingly predominant in the brain and spinal cord. The multiple dendrites allow for expanded communication, acting as network hubs integrating thousands of signals simultaneously. On the other hand, the bipolar neuron is the sensory specialist. It features a single dendritic pole entering the soma and a single axon pole exiting it. This streamlined design is built for direct, highly specific relay. Bipolar neurons are exclusively associated with afferent, sensory impulses and are located in highly specialized, delicate systems. The vestibular cochlear system for balance and hearing, the olfactory epithelium for smell, and the inner nuclear layer of the retina for vision.
There is a fascinating evolutionary adaptation known as the pseudo-unipolar neuron, heavily utilized in our peripheral sensory systems. Let's look at its timeline. In a true unipolar neuron, common in invertebrates, a single process extends from the soma and splits. But human pseudo-unipolar neurons actually begin their embryonic development as classic bipolar neurons, with two distinct projections. However, as the nervous system develops and space becomes constrained, a morphological shift occurs. The two projections slowly migrate toward each other and eventually fuse together near the cell body. This creates the pseudo-unipolar form, a single short stalk exiting the soma that immediately bifurcates into two long branches, one heading to the peripheral tissue to gather sensory data, and the other heading straight into the central nervous system. You will find these highly adapted neurons tightly packed in massive clusters within the dorsal root ganglia, located just outside the spinal cord.
Let's summarize these structures with our morphological classification matrix. The multipolar neuron has one axon and two or more dendrites. Its primary role is acting as a massive network hub, and it is typically located throughout the brain and spinal cord. The bipolar neuron has one axon and exactly one main dendrite. Its primary role is highly specific sensory intake, located in the retina, olfactory mucosa, and cochlea. The true unipolar neuron has a single projection serving as both axon and dendrite. While rare in adult humans, it serves as a direct relay. Finally, the pseudo-unipolar neuron has one fused projection that bifurcates into two long axon-like branches, with zero true dendrites emerging from the soma. Its primary role is to act as a split peripheral to CNS relay, rapidly transmitting touch, pain, and temperature, and it is classically located in the dorsal root ganglia.
Understanding this microscopic architecture is vital for diagnosing devastating clinical pathologies like multiple sclerosis or MS. MS is a severe autoimmune condition that specifically targets the central nervous system. The systemic breakdown begins with a critical immune system error. The patient's own white blood cells misidentify the healthy myelin sheaths in the brain and spinal cord as foreign invaders. This triggers a targeted, aggressive inflammatory attack on the oligodendrocytes, leading to widespread demyelination. As the thick lipid insulation is stripped away, the affected axons suffer a severe deceleration of their electrochemical impulses, and in severe cases, the nerve signals are blocked entirely. Because these impulses can no longer travel at the required speeds to coordinate bodily functions, this microscopic breakdown manifests as profound systemic symptoms. Patients experience a loss of physical balance, altered tactile sensation, disrupted vision, and increasingly impaired motor movement.
If we look at a histological slide of an active MS lesion, we can visually witness this immune attack. On the left side of the tissue, we see intact architecture. The empty spaces here, as we learned earlier, actually indicate healthy, uniform myelin sheaths that were washed away during slide preparation. But on the right side, the architecture is violently disrupted. This is the lesion, the active site of myelin loss and destruction. Within this chaotic zone, we find the cleanup crew, macrophages. Under specific stains, these white blood cells appear as brown, bloated structures. They are actively phagocytosing, meaning they are engulfing and chemically digesting the shattered cellular debris of the destroyed myelin sheaths. By studying these slides, we clearly see how a microscopic cellular pathology dictates a devastating macroscopic disease. Thank you for exploring the nervous system with Studious. If you found this histology breakdown helpful, please leave a like, subscribe for more medical deep dives, and keep studying hard.